Hi everyone, welcome back to our channel. Today, we are going to learn one of the most important topics in biology, that is biomolecules. Biomolecules are the chemical basis of life. Every living organism is made up of biomolecules, and these molecules are responsible for all life processes such as growth, metabolism, energy production, and genetic control. In this video, we will clearly understand the definition of biomolecules, their classification, structures, functions, and important scientists related to biomolecules. This explanation is very useful for degree students, PG students, zoology and life science learners, and competitive exam aspirants like CSIR-NET, GATE, and other biology exams. So, watch the video till the end to get a complete and professional understanding of biomolecules. Let's start the topic. Definition of biomolecules. Biomolecules are organic and inorganic chemical substances present in living organisms that are essential for maintaining life processes. They participate in cellular metabolism, growth, development, reproduction, and regulation of physiological functions. Biomolecules include macromolecules such as carbohydrates, proteins, lipids, and nucleic acids, as well as micromolecules like amino acids, monosaccharides, fatty acids, nucleotides, water, and minerals. In simple terms, biomolecules are the molecules that make up living organisms and enable them to function. Scientists related to biomolecules. The study of biomolecules has developed through the contributions of several scientists. In 1828, Friedrich Wöhler synthesized urea from ammonium cyanate. This experiment proved that organic compounds can be synthesized from inorganic substances. Hence, Friedrich Wöhler is considered the father of organic chemistry. Emil Fischer explained the structures of carbohydrates and proteins and introduced Fischer projection. His work laid the foundation for understanding the molecular structure of sugars and amino acids. Linus Pauling described the secondary structure of proteins, including alpha helix and beta pleated sheet structures. In 1953, James Watson and Francis Crick proposed the double helix model of DNA. Rosalind Franklin provided X-ray diffraction evidence that supported the DNA structure. Erwin Chargaff proposed base pairing rules, stating that adenine equals thymine and guanine equals cytosine. Classification of biomolecules. Biomolecules are broadly classified into two categories: inorganic biomolecules and organic biomolecules. Inorganic biomolecules include water, minerals, and gases. Water acts as a universal solvent and medium for biochemical reactions. Minerals are essential for enzyme activation and maintenance of osmotic balance. Gases like oxygen and carbon dioxide are important for respiration and photosynthesis. Organic biomolecules include carbohydrates, proteins, lipids, and nucleic acids. These are complex carbon-containing molecules synthesized by living organisms. Carbohydrates. Definition. Carbohydrates are organic compounds composed of carbon, hydrogen, and oxygen, usually in the ratio of 1:2:1, with the general formula, CH2O(N). They are the primary source of energy in living organisms and play important structural and metabolic roles. Carbohydrates are synthesized in plants during photosynthesis and are consumed by animals as a major dietary component. Classification. Carbohydrates are classified into three major types: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the simplest sugars that cannot be hydrolyzed into smaller carbohydrate units. Examples include glucose, fructose, ribose, and galactose. Disaccharides are formed by the combination of two monosaccharide units linked by glycosidic bonds. Examples include sucrose, maltose, and lactose. Polysaccharides are complex carbohydrates formed by the polymerization of many monosaccharide units. Examples include starch, glycogen, cellulose, and chitin. Structure. Monosaccharides contain either aldehyde or ketone functional groups. Glucose exists in a cyclic ring form. Monosaccharide units are linked by glycosidic bonds to form disaccharides and polysaccharides. Functions. Carbohydrates serve as the primary source of energy. They act as storage molecules in the form of starch and glycogen. They provide structural support as cellulose and chitin. They also participate in cell recognition and signaling through glycoproteins and glycolipids. Proteins. Definition. Proteins are large, complex macromolecules composed of amino acids linked together by peptide bonds. They are the most abundant and functionally diverse biomolecules in living organisms. Proteins perform vital biological functions including catalysis of biochemical reactions, structural support, transport of substances, immune defense, regulation of metabolism, and cellular communication. Scientists. Emil Fischer explained the peptide bond concept. Linus Pauling described alpha helix and beta sheet structures. Frederick Sanger determined the amino acid sequence of insulin. Structure. Each amino acid consists of an amino group, a carboxyl group, a hydrogen atom, and a variable side chain called the R group. Proteins have 4 levels of structure: primary, secondary, tertiary, and quaternary structures. Levels of protein structure. Now let us understand the levels of protein structure. Proteins are complex macromolecules made up of amino acids. The structure of proteins is organized into 4 hierarchical levels, namely primary, secondary, tertiary, and quaternary structures. Each level of structure plays an important role in determining the function of proteins. 1. Primary structure. The primary structure of a protein refers to the linear sequence of amino acids in a polypeptide chain. Amino acids are linked together by peptide bonds to form a long chain. The specific order of amino acids is genetically determined by DNA. The primary structure is the most fundamental level of protein organization because even a small change in the amino acid sequence can alter the structure and function of the protein. Scientist Frederick Sanger was the first to determine the amino acid sequence of a protein, insulin, and thus contributed significantly to the understanding of primary protein structure. 2. Secondary structure. The secondary structure of a protein refers to the regular folding of the polypeptide chain into specific patterns such as alpha helix and beta pleated sheet. These structures are stabilized by hydrogen bonds between the backbone atoms of the polypeptide chain. The alpha helix is a coiled structure, while the beta pleated sheet is a folded sheet-like structure. Scientist Linus Pauling explained the secondary structure of proteins and described the alpha helix and beta sheet models. 3. Tertiary structure. The tertiary structure of a protein refers to the 3-dimensional folding of a polypeptide chain. It is formed by interactions between the side chains, or R groups, of amino acids. The tertiary structure is stabilized by various types of bonds, including hydrogen bonds, ionic bonds, disulfide bonds, and hydrophobic interactions. The tertiary structure determines the biological activity and specificity of proteins, especially enzymes. 4. Quaternary structure. The quaternary structure of a protein refers to the association of 2 or more polypeptide chains, also called subunits, to form a functional protein molecule. Each polypeptide chain has its own tertiary structure, and together they form the quaternary structure. A classic example of quaternary structure is hemoglobin, which consists of 4 polypeptide chains. Lipids. Definition. Lipids are organic compounds that are insoluble in water but soluble in organic solvents. They are composed mainly of carbon, hydrogen, and oxygen and sometimes contain phosphorus and nitrogen. Lipids play a crucial role in energy storage, formation of cell membranes, hormone synthesis, and insulation. Types. Lipids are classified into simple lipids, compound lipids, and derived lipids. Simple lipids include fats and oils. Compound lipids include phospholipids and glycolipids. Derived lipids include steroids, cholesterol, and fat-soluble vitamins. Types of lipids, clear explanation. Now let us understand the types of lipids. Lipids are a diverse group of organic compounds that are insoluble in water but soluble in organic solvents. Based on their chemical composition and structure, lipids are classified into 3 major types: simple lipids, compound lipids, and derived lipids. 1. Simple lipids. Simple lipids are lipids made up of only fatty acids and alcohol, mainly glycerol. They are the simplest form of lipids and primarily function as energy storage molecules. Simple lipids are further divided into 2 types, fats and oils. Fats are solid at room temperature and are usually obtained from animal sources, such as butter, ghee, and cheese. Oils are liquid at room temperature and are usually obtained from plant sources, such as sunflower oil, coconut oil, and groundnut oil. Chemically, simple lipids are mainly triglycerides, formed by the combination of 1 glycerol molecule and 3 fatty acid molecules through ester bonds. Functionally, simple lipids serve as a major source of stored energy and provide insulation and protection to the body. 2. Compound lipids. Compound lipids are complex lipids that contain fatty acids and glycerol along with additional chemical groups such as phosphate, carbohydrate, or protein. They play a crucial role in the structure and function of cell membranes. Compound lipids are mainly classified into phospholipids, glycolipids, and lipoproteins. A. Phospholipids. Phospholipids consist of glycerol, 2 fatty acids, and a phosphate group. They are the major components of biological membranes. A well-known example of phospholipids is lecithin. Phospholipids form the lipid bilayer of the cell membrane and regulate the permeability of membranes. B. Glycolipids. Glycolipids consist of fatty acids combined with carbohydrate molecules. They are mainly found in nerve cells and cell membranes. Glycolipids play an important role in cell recognition and communication. C. Lipoproteins. Lipoproteins consist of lipids combined with proteins. They are responsible for the transport of lipids in the blood. Examples include HDL, LDL, VLDL, and chylomicrons. 3. Derived lipids. Derived lipids are substances derived from simple and compound lipids through chemical modification or breakdown. They include steroids, cholesterol, fatty acids, fat-soluble vitamins, and hormones. A. Steroids. Steroids are lipids with a characteristic ring structure. Cholesterol is the most important steroid in animals. Steroid hormones such as testosterone, estrogen, and cortisol are derived from cholesterol. B. Fatty acids. Fatty acids are long-chain hydrocarbon molecules that form the building blocks of fats and oils. They are classified into saturated and unsaturated fatty acids. C. Fat-soluble vitamins. Vitamins A, D, E, and K are fat-soluble vitamins and are considered derived lipids. They play important roles in growth, immunity, vision, and blood clotting. Structure. Triglycerides are formed by glycerol and 3 fatty acids linked by ester bonds. Functions. Lipids store energy, form cell membranes, act as hormones, provide insulation, and help in vitamin absorption. Nucleic acids. Definition. Nucleic acids are macromolecules composed of nucleotides that store, transmit, and express genetic information in living organisms. There are 2 major types of nucleic acids, DNA and RNA. Scientists. Watson and Crick proposed the DNA model. Rosalind Franklin provided experimental evidence. Chargaff proposed base pairing rules. Structure. Each nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. DNA has a double helix structure with complementary base pairing. RNA exists as a single strand. Functions. Nucleic acids store genetic information, control protein synthesis, transmit hereditary characters, and contribute to evolution. Thank you for watching this video. I hope this explanation helped you to clearly understand the topic of biomolecules in a simple and professional way. If you found this video useful, please like, share, and subscribe to our channel for more biology and zoology concepts. If you have any doubts or questions, feel free to comment below. I will try to explain them in the next video. Keep learning, stay curious, and see you in the next video. Thank you.